All right, this is continuation of, this is part 2, continuation of chapter 4, adaptive immunity. We just went through kind of a general overview of the adaptive immune system and T and B T cell differentiation. Now we're going to look at B cell differentiation. And very much like T cells, they develop from the hematopoietic stem cell, developing into a common lymphoid progenitor. The common lymphoid progenitor matures in the bone marrow to become mature B cells. That's different than T cells, which leave and go mature in the thymus. Instead, B cells actually are required to develop in the bone marrow, and we'll see why in just a minute. So, as I said, starts with the hematopoietic stem cell, develops into the common lymphocytic progenitor, common lymphoid precursor, early lymphoid precursor, whatever you want to call it. It means the same thing. The differentiation process is can be broken down into 3 distinct phases. The first is the antigen-independent phase. And at this point, we develop all the way from the common lymphoid progenitor through mature but naive immunocompetent B cells. That is, they haven't been challenged by a foreign antigen yet. They haven't encountered an antigen yet. So it's antigen independent. So the maturation takes place without having seen an antigen ever. The antigen-dependent phase is our actual activation of B cells once it encounters a foreign antigen. And then we can further have differentiation of activated B cells into antibody-producing plasma cells and even memory cells. So let's look at the B cell differentiation antigen-independent phase. There are actually 4 subpopulations of B cells in the development towards a mature B cell during the antigen-independent phase. We start out with, moving from the stem cell to a pro-B cell, to a pre-B cell, immature B cell, and then down here to mature B cells. We're going to look at those 4 phases of development. The common lymphoid progenitor, precursor, or B cell progenitor needs to come in contact, and so pro-B is the progenitor B cell. Pro-B B cell progenitors have to mature in the bone marrow because they require contact between the pro-B cell and the stromal cells of the bone marrow, and you can see that there's a receptor that allows for that to happen. We also have to have some cytokines, some growth factors. For example, an interleukin we call interleukin 7 is going to facilitate this maturation. During this stage, the pro-B cell, the genetic rearrangement of DNA starts to code for antibody production. We're going to be coding for the parts of the immunoglobulins called heavy and light chains. So the genes we're doing all sorts of versions of heavy and light chains. We're starting to get to that point. So we're coding for parts of the immunoglobulin, not the entire immunoglobulin itself. We're going to talk more about that in chapter 5, so I'm not going to spend a lot of time on it now, but we should know that immunoglobulins are made up of 2 heavy chains and 2 light chains. Light chains are just small chains. And put together, they make an antibody. We need to develop, just like we did for T cells, antigenic diversity. So we're going to have this random gene arrangement as well in order to code for variations of the chains. In fact, pro-B cells, we're not really coding for the light chains at this point. We're actually, we're not going to develop the final light chains at this point. And we're going to develop some alpha and beta chains that are going to be stand-in for the end product for light chains for the short term. The gene rearrangement that I'm talking about, as I said, is going to create the variable regions on the antibody chains. And it also needs to be done in a very sequential order that we're not going to focus on today, but you should be aware that if this is one of those first stops, that if we don't rearrange in a certain sequence, pro-B cells don't move on to B cells. So it's another little stopgap section for us in the maturation of B cells. cell stage begins once we see the synthesis of those heavy chains, the products of that gene rearrangement within the cytoplasm of the B cell. So once we see the development of heavy chains in the cell, we call it a pre-B cell. Let's take a look at this illustration here, and you can see we're developing heavy chains right here. These heavy chains are called mu chains. I'll talk about those in just a second. And we're also going to be developing those surrogate light chains. These aren't the final light chains, but it's an alpha and beta chain that are later going to stand in for the light chains of the immunoglobulin. Oops, here we go. The mu chains, the heavy mu chains, this stands for mu chain, is going to be the actual heavy chain that develops into an IgM class antibody. There are 5 classes of antibodies. The first one to develop is IgM, and that's going to be the hallmark at this point that we see. Those mu chains, as I said, once they accumulate in the cytoplasm, we know that we're in the B cell, the pre-B cell stage. These heavy chains can eventually bind in with the surrogate alpha and beta light chains and express on the cell surface. The heavy and light chains are what we call surrogate chains. The alpha and beta chains are what we call surrogate light chains. You have to have, cells have to have a heavy mu chain and the alpha and the beta surrogate chains in order to survive and move on. Without those, if we don't see the parts needed to make an immunoglobulin, the cell is targeted for cell death. But as long as we have the mu chains developing in the cytoplasm, we are producing alpha and beta immunoglobulin chains, those surrogate light chains on the surface, we are good to go. And we're going to see that we can move on to the next stage of differentiation, the immature B cell. The presence of a complete IgM antibody on the surface of the cell signals the fact that we got into the immature B cell stage. So again, go back here. You can see an IgM molecule here. It's tough to see here, but I think if you look in the book, you're going to see one dark greenish, I think, chain that starts at the end and goes all the way to the cell surface, and another one here. Those are the mu chains. A heavy chain is called that because it's a big heavy chain. The light chains are short from here to the junction of the Y here and here. Those are the light chains. So there's 2 heavy and 2 light chains that we see. Once we express that immunoglobulin on the surface of the cell, we're at the immature B cell stage. The mu chains are no longer detectable in the cytoplasm in an immature B cell. They're all being used to produce immunoglobulin. Antibodies have a variable region, just like T cell receptors have variability, and that genetic rearrangement caused that antigenic variability. So do B cells. These immunoglobulins right at the end here that stick out into the world on both sides of the heavy and light chain, the part of the Y here and here, that's where the antigenic diversity takes place. And so those variable regions provide different receptors for different specific antibodies or antigens. If we don't have that variability, they're not an effective immunoglobulin. In addition to that IgM developing on the surface of the cell of an immature B cell, we also start to see some other proteins. We start to see something called CD21. CD21 is a protein, a receptor for something called complement. We talked briefly about complement and how it is part of the immune system that will end up causing a destruction of a pathogen. We'll talk about complement in chapter 7, so we're not going to spend a lot of time on it now, but CD21 is a protein that develops on the surface of immature B cells, and it increases the likelihood that the complement sequence causing death of the pathogen is going to take place. So that's a helpful early protein marker of cells. CD40 similarly will be an early marker, and of course we need to start to develop our class 2 MHC molecules on B cells. B cell differentiation of immature B cells also includes kind of our test method. As we saw with T cells, we tested those T cells to see if they reacted with self-antigens and unrecognized self-MHC. And we either accepted those cells that had a responsiveness with self-MHC, an appropriate response, or we deleted those cells that reacted with non-MHC self-antigen, so we don't end up with autoimmune disease. The same process of developing tolerance and understanding which B cells are going to be great within the host's own immune system is a process we call central tolerance. And it's going to involve eliminating B cells that have self-reactive receptors and that means they have antibodies that react with self. So immature B cells that will bind to self-antigens get either inactivated or eliminated in the bone marrow. And truthfully, most B cells die this way. 90% of the B cells die this way. The host is actually, your body is pretty efficient, we've talked about before, and B cells can actually be recycled, some of them. Some die by apoptosis, some actually get recycled. I'm going to talk about the process of central and peripheral tolerance in a little more detail in another chapter. But for today, let's just remark that immature B cells are being educated, and we're looking to see which ones are going to not respond to self-antigens in a process we call central tolerance. Those tolerant immature B cells, the ones that don't respond to self-antigens, are the ones that leave the bone marrow and go out to the secondary organs. At this point, the B cells are mature, they go out to the spleen, and they'll continue to develop in the spleen. They can either stay in the spleen or migrate to other tissues. The mature B cells that remain in the spleen are called marginal B cells. We talked about how the high degree of blood flow through the spleen provides an excellent point of contact between B cells and foreign antigens. As a result, B cells hang out in the spleen, and they're allowed to quickly respond to blood-borne pathogens. Similarly, some of our mature B cells go out to lymph nodes and other secondary organs, and we call those B cells follicular B cells. These circulate through the lymphatic system and are exposed to antigens in any of the secondary tissues, the lymph fluids, for the same reason. This is where we're going to find foreign pathogens, and so we want to have mature B cells there ready to be activated. In addition, mature B cells are characterized by exhibiting IgM and IgD, and it depends on the cell. So those that remain in the spleen exhibit IgM. Those that remain in lymph nodes exhibit IgM and IgD. They don't have to stay there, though. As you remember, they can leave the tissues. Lymphs can leave the tissues. IgM, we know, is the first responder of the immune antibodies, the immune system's antibody production. They're not super specific, but they do the job sort of by brute force, and we'll talk about that. But IgG, for the longest time, we didn't really know what it was. We actually, the way that I learned to remember it was IgG was the I don't know antibody. No one really understood the role of IgD. Theories in immunology today really are suggestive that the presence of IgD prolongs the life of activated B cells. So that's the theory behind the purpose of IgD. Mature B cells are activated when they come in contact with an antigen, a foreign antigen. And once they are stimulated by that antigen, if they enter that antigen-dependent phase, here they form memory cells. They produce antibody-secreting plasma cells, and they start to have a longer life. Unchallenged B cells have very short lifespan, just days, but the stimulated B cells transform into a blast phase where they can produce, again, memory cells or start to secrete antibody as plasma cells. Once the mature B cell is antigen activated, the B cells can live for a longer period of time, 6+ weeks or more. And the reason being, we want those memories and those active B cells to be out hanging around and surveying and looking for foreign antigen to react to. So the process of antigen-antibody binding is a really important one of the immune system. We're going to see it in action in a couple of different places as we go along. But we're going to talk about the fact that, we did talk about the fact that binding antigen to antibody can, for example, block the antigenic site of a foreign pathogen so that it can't bind to a host cell, or it can signal cell death of that foreign pathogen. In a number of ways, it can also make that foreign pathogen more susceptible to phagocytosis. So this antigen-antibody reaction is really important. So let's look at what happens after we have mature B cells. They can actually further differentiate. So again, remember, mature B cells are not end cells, right? Mature lymphocytes are not end cells. They can further differentiate. And in this case, they do. They will, once activated, some of them are targeted to become plasma cells. They're triggered by cytokines to become mature B cells. And that's through the aid of those lovely T helper cells that we've seen. Plasma cells are different than normal lymphocytes. They have a lot of cytoplasm, lots of immunoglobulin within the cytoplasm, but very little surface immunoglobulin, actually none. The immunoglobulins, you can see them in these tiny little bubbles in the cytoplasm. Those little vesicles are all filled with immunoglobulins waiting to be released. Plasma cells, compared to a lymph, much bigger in size. You don't always see the immunoglobulins developing here. Sometimes you just see the plasma cell. So if you don't see the little vesicles with immunoglobulins, don't worry. Plasma cells to produce antibodies. They're found in peripheral lymphoid organs. They also can be found in the bone marrow. We see them in high numbers in a number of different diseases, as well as in response to an antigen or pathogen. And we'll talk about those a little bit through the semester, and you'll spend a lot of time on them in hematology. And the cytoplasm contains a high degree of endoplasmic reticulum. So let's look at the parts of the plasma cell. Plasma cell is characterized by this off-center, this eccentric nucleus. You can see it here. It's pushed off to one side. Some people say it looks like a clock face chromatin pattern. To me, it just looks very coarse. It's very chunky. It's not a really fine chromatin pattern that we would see in a younger lymphoid cell, for example. It's very chunky, and you can see, variations here. It's coarse. We have this really basic, this very blue cytoplasm, and it looks just like you see here. That's exactly what it looks like. And around the cytoplasm, you have this a perinuclear HOF. Around the nucleus, HOF, H-O-F. It's a clearing. Right there's the Golgi apparatus. It clears, and you don't see that dark blue cytoplasm there. And then again, you see a really abundant cytoplasm here. Bigger than a typical lymphoid cell. So if we remember, a normal length. The cell, the nucleus is about the size of a red cell. Look at a red, and then most of the cell is nucleus, and then look at this plasma cell compared to a red cell or even to another lymphocyte, considerably larger. So now that we've seen the differentiation of T cells and B cells, let's look at the roles of the cells and finish up our concept of adaptive immunity. T cells, we know the importance of antigen presentation relative to T cell activation and the acquired immune system. MHC class II molecules present antigens of an exogenous nature, those from outside of the cell, to CD4 positive T helper cells. We learned earlier how the peptide that's presented binds to that variable T cell receptor of T helper cells. And what we're going to see is that this binding of the peptide to the T cell receptor causes the T helper cell to release cytokines. And when it does, it activates the T helper cell. Once it activates the T helper cell, as we know, we can develop into a number of different types of cells. There can be a number of effects from this. Some of the things that we saw that can happen is we can develop additional CD4 effector cells. We can have a clonal expansion. We can replicate our T helper cells in order to help out with the pathogen that's been seen in the area. We can produce T memory cells. We can activate cytotoxic T cells to induce cytotoxic cellular immunity. We can enhance the humoral immune response. The activated T cells can release cytokines that trigger B cells to produce antibody. And we can release cytokines that can cause neutrophils and macrophages to develop inflammatory response of the immune system. So the same activated T cell, remember again, on what kind of an antigen it is. The kind of antigen that gets exposed to and gets presented to the T helper cell, that cytokines are released from the antigen presenting cell that's going to trigger this cell to produce other cytokines. So cytokines come from here to trigger this T helper cell. This T helper cell releases cytokines, it becomes activated, and let me start that again. This cell releases cytokines that activates this cell. An activated T cell would release more cytokines that are going to cause a number of different cells to develop. Memory cells, cells that can release cytokines to produce B cells, cytotoxic T cells, or inflammation. So those are some of the effects of antigen presentation. Some of the actions of cytotoxic T cells, I think we're pretty aware of at this point. Cytotoxic T cells, those CD8 positive T cells, when presented with pathogens through MHC class I, so those intracellular, those endogenous pathogens, or even tumor cells can be presented by MHC class I. Once those cytotoxic T cells are activated, they can move the lymph nodes and the spleen, go out and bind and kill other infected cells of the host. It's important to know that those naive T cells, though, require 5 to 8 days in order to differentiate into cytotoxic T cells. And so when I said at the very beginning of the lecture that this is not a process that is immediate response, it's not. B cells take even longer to produce effective immunoglobulins. So this isn't a fast process, and we really rely on the innate immune system, but we rely on T cells to do, we rely on the adaptive immune system to, respond when it can, but also start to produce that memory. Additionally, cytotoxic T cells, once they differentiate, will proliferate and create cytotoxic T cells very specific for that antigen. Those cytotoxic T cells, we remember, release perforins and granzymes. This is a great little illustration. So the perforins make a little pore that puncture through the membrane, the cell surface of the target cell, and create a little pore. And the granzymes, once the pore is completed, can pass through, and cause cell death. Pardon me. Sneezing here. All right. And then the role of B cells. So we understand, and we've seen a lot of the discussion about T helper cells. We understand and have seen the action of cytotoxic T cells. So how do B cells work in the adaptive immune system? There were the two mechanisms. There was a response to T-dependent antigen and T-independent antigen. And what that means is some B cells can act independently of T cell presentation or that T cell trigger, and some require that the T cell has been activated. So let's look at the two different mechanisms of B cell response to the adaptive immune system. So the T-dependent antigen response actually requires that B cells respond secondarily to T helper cells having been exposed to an immunoglobulin. Activation of these B cells takes place in lymphoid tissue, and the antigen binds to what ends up happening is you have the T helper cell has their receptor that has the little peptide in it, and then we have the receptor on the B cell as well. There's a secondary receptor, that CD40 that I talked about, that is one of the secondary markers that shows up on B cells as they mature. And you can see they bind together. Both the B cell and the T cell have a CD40 version. Once that says T helper cells have a C40L and B cells have a CD40. But they both have a CD40 marker that aids in this process. And so when we have this binding of the peptide and the CD40s on the helper cell and the B cell, the T helper cell releases those cytokines and activates the B cell to produce antibody. So that's the first way that B cells are antigen dependent. The second is that or the third, first, second, and then the third, although I'm in a different order. The third way is really that we can have remember we have surface immunoglobulins on those naive B cells. Remember, this isn't a plasma cell. We have the heavy chain and we have the light chain. And this area right here at the top of these two chains is the variable region. It's going to bind to specific antigen. So we have an antigen here with a couple of epitopes, and they bind together, and they cross-link two of these immunoglobulins on the surface of the mature B cell. This cross-linking activates the B cell to produce antibody. So we have three mechanisms. We have presentation of peptides. We have the binding of the CD40, CD40L receptors or markers, and then we have the cross-linking of immunoglobulins to activate it by binding to antigen receptors on a pathogen. So those are the three ways that the antigen-dependent response can produce activated B cells. These activated B cells then transform and do what? They become plasma cells and produce antibody. Yes? Yes. The We do not need T cells. What happens though is we have the capacity for, again, here's a foreign antigen. The antigen cross-links with epitopes, several epitopes on the antigen cross-link and bind to multiple antibodies on the surface of the B cell and causes antibody production. So this is T independent. We don't need the antigen, we don't need the T cell to do that. We also have the capacity for the antigen to link directly to a toll-like receptor. If you remember when we talked about toll-like receptors, you can have the antigen link directly to the toll-like receptor and activate the B cell. So we don't need a T helper cell in order to do that. And then finally, talking about identification of lymphocytes. Why do we care what they are? We're going to see when we talk specifically about some of the disease states associated with lymphocytes that we can identify malignancies. We can monitor immunodeficiency diseases. It's really important to know how many lymphocytes and what kind of lymphocytes someone has if they have immunosuppressive drugs or they have a disease with immunodeficiency like HIV. Then we can see how well their immune system is going to respond. And the types and numbers of lymphocytes are going to be looking in the direction of diagnosis. How do we identify them? You know, on a peripheral blood smear or a bone marrow, you can't see some of those stages in detail. Not even with like an electron microscope, you're not going to see it. So we rely now on cell surface markers. In the past, we were able to use special stains that would help us identify very young precursor cells and classify them into myeloid or lymphoid. But it wasn't always a great, or it didn't classify them the way that we can do it now, which is by looking at those cell surface markers and segregating lymphocytes into various subsets. When we can do that, we can hone in really clearly on the disease state that someone has and then how to treat it, what the prognosis is. So using flow cytometry, which we're going to look at in chapter 13, is really a very, very effective tool in identifying lymphocytes today. And I think that's it. You can look through the summary, ask me any questions you have, and I will speak to you soon. Thanks.